All right, I think we're ready. Thank you all for coming here today. I'm excited to introduce Dr. Clifford Harpole. Clifford received both his undergraduate degree and PhD in biology from the University of Kentucky, where he worked with Dr. Vincent Casson, studying the role of the pineal gland in circadian and seasonal rhythms in the singing behavior of songbirds. And in 2021, he joined the lab of Dr. Arkarab Banerjee at Cold Spring Harbor Laboratory as a postdoctoral fellow. And then some fun facts outside of the lab, he has a pet cat named Parsley and a cockatiel named Tofu. But yeah, so today he will be presenting his talk titled, I guess it's The Role of the Midbrain PAIRId in Vocal Repertoire Expansion in Singing Mice. But yeah.
Thank you, Kelci, for the introduction, and thank you to the organizers for putting this together and the selection committee for choosing me. I'm honored to be chosen, especially by fellow trainees, so I'm very excited for the opportunity to share my work. Yeah, Kelci covered everything on my first slide, but I also want to point out that the data that I'm showing you today, there's an equal co-author for it, a grad student in the lab named Mike, who all the cool stuff, or in my opinion, cool stuff wouldn't exist without him as well. So keep that in mind as I share my data, please. Yeah, as Kelci suggested or said in her introduction, um, my first love in biology was circadian rhythms and seasonal rhythms. And I also love animal behavior. So that's what I studied for my PhD. And when I was looking for postdocs, I wanted to sort of learn some more systems neuroscience techniques so I could sort of um, further my love for this. And I realized, for a few years of my postdoc here, that what I love so much about circadian rhythms is the field began by quantifying behavior very carefully, and it revealed something surprising about the brain. And I realized that's the most rewarding type of science to do for me. So the data I share with you today are sort of along those lines, I think, as well. So here's an outline for my talk. I did change model organisms from songbirds to the singing mouse. So I'll give you an introduction of why I think singing mice are also fascinating. And I'm just sharing the data from Worm project today. So I'll go over some behavior and some quantification of behavior, and then get into the neural mechanism of that behavior. And then there'll be some time for just some of the discussion as well. I don't know how to go over Zoom, but I don't really mind being interrupted. So as long as I notice it, you can ask a question in the middle, or we can try it, I suppose. But there'll be plenty of time for questions at the end as well. Yeah, so let's get into it. The introduction, why singing mouse? So when I was looking for postdocs, I had sort of a silly process. I just went to like science.org, look for jobs, and I came across this one that was studying the singing mouse. And I went straight to YouTube and I saw this video here that shows the singing behavior of a singing mouse. And given that I was familiar with songbirds and their vocal production, I also knew some mammal literature and how laboratory mice and laboratory rats tended to make ultrasonic vocalizations. So when I watched this YouTube video of a singing mouse, I thought it was very alien for this mouse to about to play it, but they make this many seconds long human audible vocalization that's very loud. And it seemed very alien to me. And I just was fascinated. I really wanted to know how the brain produces these songs and perhaps how evolution brought about this sort of behavior. So yeah, here's the behavior. The mouse on the left is singing, and the mouse on the right is in like a vigilant state. And then less than 1 second after the song on the left, the mouse on the right replies in this sort of counter singing behavior. So like I mentioned, they had these really loud human audible songs. This audio pitch has not been manipulated. And this turn-taking behavior is very robust in singing mice. So here's some other facts about singing mice, where they come from and how they live. So they're called Scotinomys teguina, are the ones that we study in the lab. They're little, they're about 12 grams. So they're smaller than a laboratory mouse. They're diurnal, so they're active during the daytime rather than nighttime, which most rodents are active during. And they live up in the mountains, ranging from southern Mexico down to Panama, sort of these mountainous ranges here. And Steve Phelps and others have have been able to set up some paradigms such that we can breed them readily in captivity. There's some evidence that these songs that they produce that last for many seconds are an honest signal, such as singing mice that were captured in the wild that sing longer songs tended to also have higher concentrations of circulating leptin, suggesting this sort of body condition can affect their ability to perform longer songs. And important for us in our neuroscience department, singing mice are experimentally tractable for systems neuroscience. So we've done silicon probe recordings in singing mice, and further, these AAVs that are developed in laboratory mice work as expected in singing mice as well. So we can do optogenetics, chemogenetics, viral expression, and otherwise. So my advisor for my postdoc, Arca, when he was in Michael Long's lab at NYU for his postdoc, he developed that counter singing paradigm that I showed you the video of, where one mouse sings, and then typically in less than a second, the other mouse will reply. This paradigm can naturally delineate these sort of sensory motor and motor epochs where you're listening to the sound, you're deciding to reply, and then replying. So you have this initiation and then the motor control side of it. So you see the sensory evidence accumulation and the decision to respond back. The vocal initiation is motor control. And singing mice is sort of an organism, gives you this opportunity where you can study the neural circuit evolution of how genomes change through evolutionary time and give rise to changes in neural circuits and give rises to these changes in behavior. So this is sort of a PI slide, but the bit of data that I'm talking to today refers to sort of the motor control and the production of the vocalization, as well as some changes in neural circuits and changes in behavior throughout evolutionary time. So let's get into it. We'll start off with some behavioral quantification. And, we had this singing mouse song. That's how they get their name, pretty well studied. But we had the question, a pretty simple question, is do singing mice employ distinct vocal modes across different social contexts? For example, when you have singing mice and they countersing, the best way to get them to countersing is to sort of have them on cabinets that are stacked on top of each other, such that they can't see each other, can smell each other just a little bit maybe, and can just barely hear each other. So sort of you have this mid-distance paradigm where they do this turn taking. What happens when you put them within a few body lengths of one another? But there's a big problem in bioacoustics, is that when you have two small animals that are close to one another, it can be very difficult to determine which animal is making which vocalization as you record from them with the microphone. And the field has a number of ways to solve this. Many researchers have like a microphone array with mini microphones in an acoustically transparent environment, and they use sophisticated analysis or a neural network in order to quantify the source of the sound. Other people have had good success by having wearable microphones on the head of the bird or the mouse. We took an orthogonal approach in order to solve this problem of attributing vocalizations to individuals. And we had two animals placed in separately acoustically dampened enclosures. Each enclosure has its own microphone and its own overhead camera. And then the animals can interact across a perforated divider here. And we called this method, just to have something to refer to it as PAIRId, which is partial acoustic isolation reveals identity. And I'll show you what it looks like when two mice interact within our PAIRId paradigm. Here you have the overhead cameras, and on the left, it's a male mouse. The right is a female mouse. Here is the output of some markerless pose tracking that we performed with the open source software Sleep. And then here are spectrograms of audio. Second. This is the left mouse's microphone, and this is the right mouse's microphone. And I'll play it. Here's what it sounds like, and the audio pitch has been reduced by 1/8. It'll be loud, sorry, in a second. Right. So this was some typical vocalizations from within PAIRId. As you see, they make a lot of quiet vocalizations, and then they also continue to sing when they're in these close by social settings. So they had the very loud stuff was what we classify as their songs. You also may have noticed that we have these blue and orange highlights on the different vocalizations. This indicates that that vocalization has been assigned to that individual given our, sorry, given our assignment algorithm. So the way that our assignment algorithm works is that we run detections on both microphone sets of audio. And then in 80% of cases, a vocalization is only detected on one of the microphones. In that case, if we get it for free, it's very easy. 80% of the vocalizations, due to the partial acoustic isolation, get assigned for free. And then in instances such as this strip of vocalizations right here, where there's sort of an overlap in detections, we have an algorithm that sort of compares the shapes of the spectrograms of the two audios. And it can determine if the mice just happen to vocalize at the same time, like in this example, or if there was acoustic bleed through where we can ignore it on the quiet side and keep the loud side. And our assignment algorithm can take it from 80% to 98% of vocalizations. It's also highly accurate. We have a 0.87 F1 score. So we're quite confident when I show you vocalizations of a single individual that those are attributed correctly. And here are some vocalizations from the PAIRId setup of one individual mouse. Each strip here of this raster plot represents a single individual vocalization, and this is just 30 seconds of time. So they're highly vocal within this setup. And you might notice, as we did, that there are periods of time, such as right here, where the vocalizations seem to have a temporal structure, and they're a little more stereotyped, and some less so here in these examples. So we set up a procedure that looks at the acoustic structure such that we can categorize vocalizations. So we sort of have a multiple step process, and the output of this process produces two main categories of vocalizations. One are these temporally structured ones that are their namesake songs, and then some extra vocalizations shown here in pink, we classify as USVs or ultrasonic vocalizations. And these individual arrows sort of indicate some examples that I'm about to show you. And here's what those vocalizations look like in spectrogram form. Here is a spectrogram of a singing mouse song where you get fast notes at the beginning, slower notes at the end. And then here are these example of the USV spectrograms from the individual arrows of the raster plot. So if we look at all the vocalizations, we have a data set here of 10 mice across 60 hours. And when you look at the acoustic properties of these vocalizations, songs differ greatly from USVs. So songs are on average 30 decibels louder, they're 10 hertz faster, and these fast beginnings can be more than 20 to 25 hertz for singing mice. And then also the bout duration. They string together many more notes in songs, and on average, they're 4 seconds longer. So given that we have these vocalizations here, we wanted to place them in a phylogenetic context. So I already mentioned the term USVs a couple of times. And USVs are very widespread among rodents, specifically in the families of Muridae and Chrysetidae that singing mice and laboratory mice are from. And actually, representative species of each subfamily of those two big families of rodents have been shown to produce adult USVs. So given the widespread nature of this, we picked laboratory mouse due to the fact that they're very well characterized, kind of convenient. So we recorded some USVs from laboratory mice in our lab as well. And I'm going to superimpose those on these CDF plots here with this gray line. And as you can see, the gray line is highly overlapping with the pink line, which is the USV line, showing that in regard to these acoustic and temporal properties, singing mice and laboratory mouse USVs are highly overlapping. I'll also accumulate some extra evidence throughout the rest of the talk, but we're very confident that these sort of quiet vocalizations that singing mice that produce due to these properties and some extra evidence that we accumulate, are homologous with the USVs that are widespread across many rodents, and therefore represent sort of this ancestral vocal mode that singing mice have held on to. I'll talk more about that in a little bit. But given that songs and USVs are so different in these acoustic properties, we had a hypothesis, and that was that they would be used in different modes, given how different they are in these acoustic properties and different social contexts. So that was the next experiment. Um, we had singing mice in PAIRId with a partner, as you've already seen. And then we also had a setup that the day before we put them in PAIRId, we split up each individual mouse and had them separate and recorded them when they were alone. This is sort of a proxy for, of course, being in PAIRId, they're within a few body lengths of a conspecific. So this is sort of represents a close range social interaction. And when they're alone in their box, they can't smell any conspecific, can't see any conspecific or hear any conspecific, sort of represents having a conspecific at an infinite distance. So when a mouse is alone, it continues to sing quite robustly, and the primary vocalizations that they produce during this time are indeed songs. So they sort of only produce songs, or at least primarily produce songs when they're alone. And when you bring them close to a conspecific, you see that their repertoire complexifies and they start producing all different kinds of vocalizations, as shown here in this raster plot. These are 30 examples of some highly vocal sequences across our 60 hours of data. I also want to point out sort of a little detail that's one of my favorite details from this data set. Each line here represents the vocalizations of an individual mouse. And then you can see that quite often they'll go from making USVs to making songs in these very rapid and smooth transitions. A little bit more on this later, but this is one of my favorite details, are these really interesting transitions that they make when they're in this sort of setting. But given this and the differences in acoustic properties, we conclude that singing mice have two dominant vocal modes that are used in different social contexts. So songs are primarily used for long-range communication, given this alone data and some data from the literature as well. And then USVs are primarily and greatly enriched when they're close by to a conspecific.
Liv, can I interrupt for a
Yeah, please.
clarification question?
Yeah,
real
sure.
quick?
Yes. Mhm.
Two things. One, what are the dark blue? Let me turn this off.
Yes.
So what are the dark blue ones?
Yes, that is a rare vocal type that we call semi-songs. It represents just like 5% of the vocalizations. I can show you some spectrograms at the end, but it's sort of these vocalizations, they're a little bit faster than USVs, and they tend to have one or more loud notes, but it's sort of something in between a song and a USV.
Okay.
They're pretty interesting, and it's another evidence that I can bring up later now that you brought it up, that there's something kind of related between USVs and songs, but these are sort of a rare one. However, it looks pretty enriched on these really hot examples because, very common, they tend to make some of these semi-song sequences in advance of a song. So,
Okay.
they're only 5% of the data set, so it's hard to say too much about them, but they're pretty interesting. Yeah.
And then maybe the second question. Oh, I'm getting echoed, sorry. How many patterns of songs do we have? So if you classify the songs, how many patterns would we see?
Yeah, I'm getting into that in like 2 slides. The answer is 1. They're very stereotyped, both within and across animals. And yeah, 2 slides will show you a lot more about it. And it's one of the really interesting parts, in my opinion.
Sounds good. I can wait 2 slides.
Perfect. Yeah, great. So yeah, just to quickly summarize, that there are 3 key behavioral innovations between songs and USVs that are needed for songs. One is that they're much louder. Songs are much louder than USVs. It's shown sort of this trace here. Songs also can be much faster than USVs. Showing this trace here, it can be 20 to 25 hertz. They don't do it reliably when they're in these close by settings, but when you have them around 4 meters apart, like I discussed before, they'll do this turn taking with songs. So this is sort of a third innovation. And as Ali brought up, sort of songs are also very stereotyped, both within and across individuals, where you very often see fast notes at the beginning. I mean, exclusively see fast notes at the beginning and slow notes at the end. Um, and we were always struck by this, that particular song all had the same patterning across the entire species that we've ever measured. So we sought to make a linear model to capture this sort of stereotypy that Elise sort of keyed in on, wherein they have very fast notes at the beginning, and then it's basically the same note that's repeated over and over again. And with each repetition, it gets regularly and reliably longer and longer, such that if you take instantaneous note rate, which is 1 over the difference in 2 consecutive note start times, you get sort of the amount of notes per second in hertz. If you plot instantaneous note rate over note index, you get this linear relationship, such that with each repetition of the note at a linear scale, you get a longer and longer note. So you can represent a particular song with just the equation for a line, y equals mx plus b, or however you learn this back, whenever in middle school. And if we're right about this, if this is truly the way to pattern songs for all individuals across all mice, then we can integrate over this slope and come up with this formula such that we can apply this linear model of song rhythm to all songs from our data set and then predict how long the song is from note index space to time space. And when we do so, we get this really tight fit to this line, a Pearson correlation of 0.97 here for all 10 mice that we measured here, and a bunch of other mice we did just to, sanity check ourselves. This really captures, I guess, 97% of the variability of song pattern across and within individuals. So we're very excited by this because this sort of sets up a way that we can capture the dynamics of song changes sort of as variability. And then if we start perturbing the brain, we can start to see how the songs degrade. And we'll do that sort of in my last data slide. So given, let me give you another interim summary. I've been doing this a lot, but here are the big takeaways from the behavior part. Is that, given our description, we conclude that singing mice have 2 distinct vocal modes, songs and USVs that they produce. And then we have this linear model that hadn't previously been noticed before that can robustly capture the patterning of a song with just the 3 parameters of a line. And then the reason we're excited by these behavioral results, and then we'll try and take advantage of this blue text sort of in the last half of the talk here. Given this first one, that songs make USVs, this means in the same individual, we have sort of the singing mouse version of an ancestral vocal mode, and then their newly evolved song. So in the same individual, we can compare and contrast these USV mode, which represents an ancestral form, and a newly evolved song. And then given our linear model, we have a principled framework such that we can investigate the neural mechanisms of motor production of a singing mouse song. So try and come back to this in the last bit of our data. But yeah, let's get into the neural mechanism, and we'll start by, given that they have these 2 distinct vocal modes, we next asked a question. Do these distinct vocal behaviors emerge from separate neural pathways, given that they're so different from each other in loudness, tempo, stereotypy, bout duration, etc.? It could be possible that they just have these parallel motor pathways. But given these little details, that there's the fast and smooth transitions, there's that 5% mode that we talked about briefly of semi-songs, that's sort of something in between the songs and USVs. This could suggest that maybe they sort of share a common motor pathway, and it just operates in a different regime in order to produce a song. So we asked this question, and we decided to look at this question at several different points along this vocal motor hierarchy that we introduce here. And we'll start at the bottom and look at the peripheral sound production mechanism and move up, comparing and contrasting songs and USVs in the same individuals. So let's start with the periphery here. Rodents produce sounds in their larynx in 2 different ways. They can make vocal fold vibrations, sort of like human speech, where the vibration of the cartilage in their larynx is the source of the sound. They can also do this kind of weird thing where rodents make whistles within their larynx. And this sort of aerodynamic property is what is producing the sound. And it's been known since the 70s that you can distinguish these two vocal modes by changing the density of air in which the animals vocalize. So we used 20% oxygen and 80% helium and tested this. And then our expectation was if the vocalizations were produced from a vibrational source, the fundamental frequency would not change. You just sort of see some subtle differences in the amount of acoustic power that's in the harmonics, and then the pitch of the acoustic of the harmonics will also increase. So when a human sounds like a chipmunk, it's actually these harmonics that are the cause of that. The fundamental frequency of human speech is not that affected by helium. And then if it's sourced from an aerodynamic source, the fundamental frequency would be completely dependent on the density of air. So the fundamental frequency will jump. And we looked at singing mouse USVs and sort of I let them vocalize in air, and then I pumped in this heliox gas that's 80% helium. And we saw that the fundamental frequency jumped by 1.3, roughly, 1.4, roughly. So USVs, as is common across all rodents that have ever been measured, are produced by one of these laryngeal whistles, this aerodynamic form. But I wasn't sure what songs would be like. Given that they're so much louder, it's possible that maybe they're from a vibrational source. But in these same sessions, they also sang, and the fundamental frequency of songs jumps. So songs are also from sort of a whistled peripheral mechanism, and the ratio of these in helium-oxygen mixture versus just atmospheric gas is the same as USVs. So they sort of share this whistle production mechanism. Next, we moved up the vocal motor hierarchy here, and we looked at vocal respiratory coupling. So to investigate this, we installed a intranasal thermistor such that we can measure the temperature of air exchange in the nostril of a rodent. So you get sort of the decrease in temperature as they breathe in sort of the cooler, relatively cooler atmospheric air. And then as they exhale, they have the air warmed by their body. So you see an increase in temperature and you get a proxy for respiration. And what we see here is that, each note for a song and a USV as well is produced on an exhalation. And each gap of a note has at least a partial inspiration in between it. This is true for even those fast notes at the beginning. They're not taking a big breath and making 5 notes, for example. It's all 1-to-1. So when we show a song and we show these song patterns, that's actually the rhythm of respiration itself. And this is true for USVs as well. Here's sort of a summary plot of all notes that we measured with this thermistor mouse, showing that the beginning of phonation and the end of phonation all occurs on the expiratory phase. Okay, moving up the vocal hierarchy to the final point, which is vocal gating. We wanted to go into the brain. So the place that we looked for the rest of this study is the midbrain periaqueductal gray, the PAG. And this has been well established as a critical locus for innate behaviors, including vocalizations, across vertebrates. Even vocalizing fish use the PAG as part of a gating mechanism for their vocalization. So it's highly conserved, and more specifically, it's been well established that sort of a particular part of the PAG, this caudolateral PAG, clPAG, that these neurons in this column of the PAG project to hindbrain phonation circuits, such as the nucleus retroambiguus and the pre-Bottinger complex, and that these neurons, in the laboratory mouse at least, are necessary and sufficient for USV production. So we decided to look at the role of the homologous part of a singing mouse brain, their clPAG, to see, how USVs and songs are represented here. So for this video I'm about to show you, we did unilateral excitatory optogenetics in the homologous part of the brain for clPAG. And I'll show you this text will turn blue when the laser turns on. Yeah, so we turn the laser on and the mouse makes USVs. is basically the point here. This was true for all 4 mice, that for the entire duration that the lasers turned on, they continue to vocalize. And importantly, all of these evoked vocalizations shown here in this bright blue trace were highly overlapping with naturally elicited USVs, nothing related to songs. So the songs is in this like teal or cyan trace, and the blue and the dotted magenta represent sort of fictively evoked and naturally occurring USVs. Nothing close to songs here. So does this part of the brain do anything for songs, was what we asked next. So to investigate this same part of the brain, this time we bilaterally injected tetanus light chain toxin to synaptically silence these neurons. And when we do this, the mouse goes mute. So we inject, and then it happened very fast, like within 2 to 4 days, the mouse stopped singing and also stopped being able to produce USVs. I sort of alluded to this earlier, but given that songs have this stereotyped patterning of fast at the beginning and slow at the end, we had the hypothesis that given this temporal structure, we could do something kind of unique with the singing mouse songs, which is after injecting, but before going mute, we can look to see if the songs deteriorated in sort of a principled way and if we could learn anything from that. So that's what we did next. Here's a spectrogram and some loudness traces and then some note rate traces for this song before the mouse was injected. And then we injected the mouse, and before going mute, it continued to sing in this interim phase, and we get these intermediate songs as they're sort of on the path to mutism here. And this intermediate one is a little less loud, and it's a lot shorter than the blue trace. And then it's sort of the last bits that they're able to spurt out is this represented in this green trace. It's quite a bit quieter, and the song is much shorter. And this is true for all 5 mice that we did, where you see this gradual loss of loudness as they lose their ability to sing. And then the songs also get shorter and shorter as the sort of virus continues to express and express its functional protein. If you look closely at this instantaneous note rate graph, here we sort of notice something about these plots, and I made a little animation to sort of illustrate what we noticed. That sort of, here's a typical trace in cartoon form, and then as the virus expressed itself, the song got shorter and shorter with each sort of measurement, such that you get this final short song here. And then we notice that it's sort of, ignore that I splayed these out. The slope isn't that different, but I'd sort of splayed these out here. But notice that it gets different by sort of reducing from the right side of the plot here, and that mice are still very capable of producing the fast notes at the beginning. It just seems like the songs are becoming truncated. And if that were true, we would see sort of a gradual increase in stop rate as the stop rates of this particular songs become truncated, and we lose the songs from the right side of this. So this would be a great opportunity to apply our linear model to all these perturbed songs, which is what we did for the final slide here. So given that observation that we had a hypothesis that song duration would vary inversely with the negative logarithm of stop rate, meaning that shorter songs would have a higher stop rate. And that is the case here. So this is sort of the black line sort of is the projection of our prediction of how this would fall. And then each gray dot here are the particular songs of these natural and perturbed mice as they sing their songs, such that shorter songs have a higher stop rate. And that's the case. And if we color code these gray dots, given the sort of degradation from blue to orange to green, we see that that's exactly what happened is across all 5 mice that we did this in, we indeed see this sort of gradual decrease in the duration, and we also see a gradual increase in the stop rate, showing that all of these songs are becoming truncated, and that the clPAG has sort of some control over the stop rate parameter of songs, sort of maintaining this long pattern. The mice become less and less capable of producing the long, loud notes at the ends of songs. So next we asked, is this just a weird thing with this virus? Is this just a weird thing with this protein? So we had the observation, and this is held up in the literature as well, is that female mice sing shorter songs than male mice. So we had the same question with these naturally occurring male versus female differences. What explains this difference in song duration? And it's the exact same thing that naturally occurs here, such that male mice songs tend to be longer because they're capable of producing these longer, louder notes at the ends of songs that females are incapable of. So it's possible that the clPAG is actually underlying this natural variation as well. And that's sort of the final takeaway here, is that caudolateral PAG is a locus that controls natural variation in song length via the stop rate parameter. Yeah, and that's it for the data for this talk, but we can discuss some here. So what I showed you as a summary is that between USVs and songs and singing mice, there's a lot of stuff in this maroon color that are shared, where they sort of have a whistle mechanism in the periphery. They're all produced on exhales with gaps being formed by inhales, and they both require the PAG in regards to their production. They're both necessary for PAG, but this caudolateral PAG is only sufficient for USV production, because when we excite those neurons, only USVs come out. So from this and other data, we have these behavioral data showing that there's these three key innovations that have evolved for songs, and that somewhere this amplitude modulation to make songs much louder, frequency modulation to have this pattern and this fast rate at the beginning, and the ability to take turns when they're in those specific settings must converge at this caudolateral PAG in some form. So that's where we are. We sort of have one part of the brain that is maybe important for the stop rate parameter. And now the search is on to see where in the singing mouse brain, where has it evolved to produce the rest of these parameters. And we have some clues from prior research where some of these parameters may have come from, and we have some ideas for future ones as well. And that's what I'll go over now. I already brought this up, but given this turn-taking behavior that I showed you the video of, where one mouse sings or you do an audio playback and the other mouse responds, Arca from his postdoc has evidence that the orofacial motor cortex is important for the ability for it to take turns with songs. So Arca and colleagues at NYU identified the singing mouse orofacial motor cortex via stimulating cortex and finding where the digastricus muscle had an EMG recording for the opening of their jaw. And then if you use Musimol to inhibit this, there exists a Musimol protocol where singing mice can still sing. So this, OMC is not required for song. But if you inhibit it, they lose their ability to respond socially and be socially stimulated by songs in these turn-taking events. So it seems like throughout evolution, the OMC has perhaps come online and to allow this sort of turn-taking behavior. And further, it's possible that the OMC also affects slope, and that could be revealed by this cooling experiment that Arca did, where he used a Peltier cooling device and lowered the temperature of the OMC. And in a temperature-dependent manner, if you cool the OMC, you see that songs get gradually longer and longer in relation to how cool and how slow, presumably, the neural dynamics of the OMC is. And sort of an interesting contrast across different evolutionary taxa here is if you cool the HVC of a songbird, which is one of these song-controlled nuclei of a songbird, the songs also get progressively longer and longer as you cool this sort of critical loci. But in contrast, for a zebrafinch song with a cooled HVC, you see that the notes get more and more stretched as the HVC gets cooled. And then as you cool the singing mouse song, they get longer and longer via adding notes rather than sort of stretching individual notes. This is just one figure from these papers. It's pretty convincingly displayed a bunch of different ways, but yeah, this is kind of a fun difference. So this suggests that maybe OMC has some control over the slope of the song and this turn taking, but sort of where does the pattern itself come from is a question that I would like to find. So my project right now is following up on some comparative transcriptomic work from our lab. Emily has a bioRxiv paper where she used MapSeq on both singing mice and laboratory mice. And one of her main findings was, I mean, her particular main finding from OMC was that in singing mice, when compared with laboratory mice, you see sort of a threefold increase in projection strength from the OMC to sort of a part of the auditory cortex that she calls the auditory region, or AUD-R here. And then of interest to me, who likes motor production stuff, you see this threefold increase to a part of the PAG from via OMC and singing mice relative to laboratory mice. So my current project in the lab is looking at this OMC recipient part of the PAG. It seems to be, it's like, 800 microns or so more anterior to the part of the PAG I just talked about. So it seems to be a different part of the PAG. It's in a different column. So I think it's a different part of the PAG. So I've been using some pathway specific, using AAV1 to deliver Cre to particular neurons and try and manipulate and silence neurons that are OMC recipient PAG. And I don't want to show any data because it's still preliminary, but these neurons seem important, and they seem to break in different ways than the caudolateral PAGs. So stay tuned. I'll tell you what these neurons do hopefully soon. But all in all, this sort of sets up our ability to answer these 4 questions of ethology as proposed by Tim Burgeon in the 60s, where in order to fully understand a behavior, you have to look at it from several different directions. And as a lab, we're doing that currently. Today, I sort of gave you the beginnings of some answers in regards to the mechanism of how do singing mice vocalize in different social environments, up at one particular part of the neural circuitry underlying their vocalizations, and then a little bit about the phylogeny, when and how do these vocalizations evolve. I have like one more slide on that as well. And we have another person in the lab named Yaman who's doing some modeling of sort of when the singing mice sing during the day and how they use their song timing during these turn taking to sort of get at what's the utility of song a little bit. And then we also have a grad student in the lab named Christina who is looking at the ontogeny of these vocalizations. She has some really beautiful data of like how this line structure develops throughout the ontogeny. It doesn't come from anything, from nothing. So sort of how they vocalize as infants to juveniles to sort of the fully formed adult song. And that will be great when she comes around to that. And given that you're in the department with Kathy Marler, I wanted to talk a little bit about some of the singing mouse relatives just here. I made this crazy tree that has way too many species on it, but I did this to illustrate like how widespread USVs are across these various rodent taxa. Pink means it's been shown in adults. So this is why I'm so convinced that, version of USV was likely shared by a common ancestor here. But the singing mouse song, lasts multiple seconds. It would be crazy if it just evolved all of a sudden. There has to be some sort of intermediates. So sort of the question is, what did this common ancestor of, singing mouse and a bunch of their related species, including Peromyscus, what was their behavior like? And if you look at some vocal modes in addition to the USVs, that's quite common among this sort of subfamily here. That includes an extra species of singing mice, these harvest mice, Peromyscus, and these grasshopper mice. They have an extra mode. These are all the whistle mechanism ones. These have an extra mode that they'll also open their mouth. I'll show you this picture here. They'll also open their mouth. They also will do sort of the posture. So it's sort of enticingly perhaps common to some intermediate form of the song, but it's a little mysterious because it has the vibrational mechanism rather than the whistle mechanism. But I think this is pretty interesting. And then given that I love circadian rhythms and that's my first biology love, I'm also find it very curious that to the best of my knowledge, all of these other rodents are nocturnal, and then singing mice and maybe their tribe mate Bayamus have recently flipped to being diurnal. So perhaps that contributed to their elaboration in some way. This is sort of a speculative slide, but I wanted to talk about these Peromyscus, the that Kathy calls SVs, the sort of extra mode in addition to USVs, that is also shared in the same subfamily here that I think is fascinating. Yeah, and that's it. So here's the acknowledgement slide. Please remember that Mike is the equal co-author here. Also, he's graduating next year, so you should recruit him as a postdoc if you're hiring. And then these are the other co-authors on this project and everyone else here. And yeah, thank you so much. I'm happy to take any questions or discussions or otherwise. Yeah.
Thank you so much for giving the talk. I think I was just gonna start off the discussion with a question.
Sure.
So, and my question relates kind of to the social context. So, do potentially repeated social interactions change like the relative use of songs or USVs, and kind of maybe does that change their function or like kind of information that's being conveyed? Oh.
Muted. That's a great question. So this is sort of a weird detail. It's not that important, but we collected these data in cohorts. So we had sort of like 3 male mice and 2 female mice, and we did all the opposite sex pairs. So the female would get like multiple males, but she would be used a little bit more in these cohorts. So, whatever. I've done the same mice and this pair ID setup, multiple times a week. And they seem to like not necessarily habituate to particular partners or something. So they seem to be pretty stimulated to make these USVs when they're in close range, independent of like recent or prior exposure. So we don't see much with the USV mode in regard to being repeated. But when one does these turn taking, I don't have too much data on it, but when you do this counter singing turn taking behavior, we tend to start them in the morning, and one mouse will sing and the other mouse will reply, and you get these tight coupled relationships. But this sort of their ability to respond, the probability response, sort of decreases throughout the day. So it seems like they sort of habituate to a particular partner in regard to their singing. um, so we always took that as sort of light evidence that maybe there's a potential to have some individual recognition in some of these song parameters. And there's like a recent preprint from the Michael Long lab with his name's Yuki, a grad student with Mike Long, and he's done some statistics showing that sort of some acoustic features in their songs and then sort of their response probabilities if they respond quickly or if they interrupt or something. Given this, he's sort of used like one of those supported vector machines or the SVMs or whatever, to show that the within songs there exists the potential to have individual recognition there. Um, so it's possible that sort of repeated, they can tell each other apart from their songs, and maybe less so these USVs. Did I answer your question?
Yeah, I think also following up on that,
Yeah.
are some songs like more consistently complex? Like, is there like variation within the songs?
Oh, yeah. Yes. So songs are typically 6 to 7 seconds. Something I sort of failed to say. When they're really stimulated, and especially in that counter singing paradigm where they're responding, they can double the duration of their songs. They can be 15 seconds, 18 seconds. So this like ability to add notes and make songs very, very long is really a strong suit of a singing mouse. So the duration is a big difference. But in the other acoustic parameters, like they tend to all have the same negative slope where they have fast notes at the beginning and slow at the end. That's very consistent in regards to like complexity and stuff. And they can be a little bit some louder than others. But there's not too much dynamics with how loud they are. They tend to be just sufficient, the singing mouse song version of loud. Um, there's some weird edge cases, maybe in 1 or 2% where they'll make like a little pulse of a song. But they're sort of rarities, but they can kind of misfire with songs. But otherwise, they're big sort of dials that they have is how long the song um, is the big thing that they have control over in order of varying and stuff.
I'm gonna open it to the group now.
Yeah, should I start?
Oh, yeah.
Or I can see it too. Makes sense.
I've turned mine so I can see.
Okay, okay.
Somebody needs to mute.
The song of our people.
I just heard it. Okay.
You might have
Can you hear me?
Yes.
Great. Have you looked for individual differences in songs? Is there any evidence of them trying to broadcast like individual information like you find with some monkeys?
Yeah, it's still, you know, I asked Kathy this at a meeting that we were both at last summer, but it seemed to be very hard to do field research on rodents. So in the field, most of the sort of outdoorsy type work, they would capture the rodents, do something with them in the hotel, and then re-release them. And then we have these sort of outbred colonies. So that's kind of most of the data we have are sort of these more controlled, you know, indoor cage type environments. But more and more, we're getting like little bits of evidence of what sort of information is contained in song. So I mentioned it very briefly in the introduction, but you know, you capture mice in the wild, and the mice that tended to sing longer songs have more circulating leptin. Same is true if you supplement exogenous leptin. It sort of affects some of the characteristics of the song. And it sort of sets up this thing of like, these songs are honest signals, and I think the ability to make the fast notes at the beginning and then the stuff like I was talking about here, the long loud notes at the end, I think that you have to be in a good strong body state in order to produce these sort of hard parts of song. So I think there's got to be some kind of selection, sexual selection or otherwise, going on, the ability to make long songs, make songs with big loud notes at the end, make fast notes at the beginning. So I think at the very least, they're kind of advertising some sort of body condition by their ability to make these loud songs. And they kind of, when they do this counter singing behavior, it's hard to tell what it means, if they're being nice to each other, if they hate each other. But they tend to start showing off with longer songs when they're doing these counter singing exchanges. And I think that's sort of the real display of sort of like pick me versus the other guy type thing. I could also have an extra slide if I can find it here. Also shows that like these singing mice are also insectivorous. So that comes up with these sort of overlapping ranges like this. So I always imagine their songs having some function. And songbirds has been shown really well that it's like, it's very energetic. It saves a lot of energy if you know your neighbors. So I imagine that that might be some component of the individual parts of song, is you like, oh, this is my southwest neighbor or whatever. And then if you get an invader that's not one of your neighbors, maybe you have a more aggressive response. This is extrapolating from songbird literature. But I imagine that they're familiar with other singing mice in their areas, as they sort of have these ranges where they hunt insects and stuff. But I would love to go to Costa Rica, which is they live in a very great place too. So yeah, these are some really great field questions that I think are really important if we try and say, you know, keep us in check of what these songs mean, if we try and extrapolate further from them and stuff. I think it's important to get to the function of it and what information is contained in it.
I have a question. Hi, Cliff. Great talk.
Thank you.
I just wanted to ask a little bit more about the potential differences in social interactions after your tetrodotoxin intervention. You've mentioned that across different animals, and especially across biological sex, these songs change quite in a very clear categorical way. As these animals started to transition to shorter and etc. songs, did their partners in the songs or other interactions change, and how did that work? Because that must give you a lot of insight into what that song really does, right?
Yeah, that's a good point. The short answer is we don't know. Sort of, you've honed in on something we made this this behavior paradigm, and then we just focused on the vocal production of one of the mouse and kind of ignored the whole interaction side of it. Like you said, that could be really rich, and it could be something that this setup that we made, this PAIRId setup, is really uniquely poised to do, but we didn't really look at it. But yeah, like you said, how do they respond to these? They don't reliably do the turn-taking behavior. That would have been amazing if they sort of would reliably do this turn-taking behavior in the close range settings, such that as their songs degrade, maybe you see like a drop in the probability of response, but that didn't happen. They don't reliably turn take when they're close by. I guess, I manually curated a bunch of these data. They're a real pain to get. So I sort of had the impression that as their songs get less loud, maybe easier seen from the slide before. Oh, too many animations. As the songs get less loud, it wasn't enough for us to analyze, but I did notice that they would essentially get interrupted by the female partner a little bit more. But that's kind of the only thing I noticed is like, they would sometimes make these quiet songs, and then you can kind of tell that, I mean, I'm anthropomorphizing, but maybe they weren't happy with the song, and then maybe they would start with a slightly louder one later. So it seems like maybe, this is just some anecdotal stuff. The real answer is I don't know, although that would have been interesting to analyze in regards to these degrading songs.
Question. So the counter singing in male and male females, both scenarios.
Yes, it's most reliable in male, but there is one paper where they tried same sex and opposite sexes, and it seems like it does exist across all, but I think it's most probable in male, if I remember that paper correctly.
So an obvious comparison would be the singing behavior between those two scenarios, because one's likely to be aggressive, the other one's likely to be choice.
Yes. Yeah, I agree. Yeah, the most reliable counter singing is male. But females do sing. As you know, I showed you in the next thing. It's kind of a cool thing. It's very common in at least songbirds in the tropics. Singing mice. Our colony originated from 9 degrees north, so they're sort of toward the tropics. For whatever reason, in the tropics, there's more female competition across animals. So the females, they sing a little less, and yeah, as I said, their songs are shorter, but females do reliably sing. But in regards to this sort of turn-taking behavior, it seems to be less probable than in the males.
But sorry, is there courtship turn-taking then?
We haven't really looked. So, like all of the data that I showed you are for male-female pairings. But if you put two females together, they also make USVs in songs. So, given that we had a divider, there's a little, I mean, the only kind of evidence I can point to is that with a divider, it seems to increase the probability to vocalize, because if we have the two mice together undivided, they'll do lots of allo grooming behavior and stuff, which is a very strong thing that singing mice do. Of like the male pins the female down and like rubs his hands on her, and he rubs them on his subventral sebaceous gland and then rubs oil on her. So it seems like if you exclude these behaviors that require touch, that they're more likely to vocalize, which was great for us as well, because we're mostly interested in the vocal production. And it seems like this PAIRId thing, we get slightly more than when they're together. But, I would have data, when I got my helium data, they were undivided because you get good vocalizations that way too. So that's it. That's all I can say about courtship is like, they also seem to be slightly pickier than laboratory mice. When I put two lab mice together to make USVs, they're also undivided. And there's often mounting attempts. Singing mice, it seems like the females are more, they pace things very slowly, and you see a lot less mounting attempts in these undivided. But it's not something I ever quantified. But they're harder to get to breed, and they seem to sort of mount less readily. So maybe there's something slightly interesting going on with their like breeding strategy or something.
And I was wondering about the PAG, so, that's associated with aggression as well. And you're not getting a lot of variability in song characteristics necessarily with the PAG simulation, right? So I
Um,
wonder whether if you gave just a slight, and I know you sort of said far distances versus close, but I wonder if you gave a subtle cue, just like scent of another male or female or whatever, cotton ball, whether you would still get these long distance, but you might be able to find some plasticity in the vocal.
yeah, I mean, the average duration of song did go down, like they went from, I mean, they eventually went mute. So eventually they made these really terrible things that are just a few notes. But it went from, typically, let's say the means like 10 seconds, to like a 4 second song. So, we lost more than half of the duration. So we see, the variability was pretty stark of, they went from quite long to not long at all to nothing at all. So we did have some variability in here, but yeah, I see what you mean. We've tried some stuff with female urine. It doesn't seem to be as strong of a cue to vocalize as it is in laboratory mice, but we didn't try very hard. And trying to collect urine from females, I got bitten a lot. So we were, we just played around with that for like a week and then gave up pretty fast. So you shouldn't believe me. But it seemed it wasn't very promising either, despite getting bitten. Yeah.
There's a oh yeah, there's a hand in the Zoom chat. You can go ahead and unmute.
Sure.
Okay, Kathy mentioned the PAG and aggressive behavior, and I know there's the literature on the columns in the PAG and how they, like studies in cats and primates, and I think guinea pigs, looking at the different columns and how they regulate different sort of vocalizations that reflect different affective states. And so I was wondering if
Yes.
this part of the PAG, if maybe the USVs and the songs reflect a similar maybe contact call, but what would you expect if you looked at maybe alarm calls or something that is in an aggressive context?
Yes, that's a good question. Yeah, PAG is very heterogeneous, but if you do a search for it, the first thing that come up will be aggressive behavior. And then second would probably be escape behavior, and maybe third would be these vocalizations. But yeah, you get these heterogeneous columns that sort of extend anterior to posterior. But we just did sort of this course, at the cell at the level of just the whatever we can inject. That's the only level that we have. Katie Cheetah had some really cool, when she was doing this in laboratory mice, she could use some really cool activity dependent ways of isolating particular vocal neurons in these pathways. And then so such that when she turned her laser on for optogenetics, the mouse didn't run around, it just vocalized. So, at the very least, we have this sort of heterogeneity that we turn our laser on, we do get the USVs, and that's what we mostly measure, but we also get this running around behavior that she was able to isolate out in a very amazing way. And then, yeah, in regards to aggression, just anecdotally, we've switched to muting a lot of mice and using those as sort of social stimuli as sort of a cheaper way to tell which mouse is making which vocalization. And in these muted mice, we get some heterogeneity. This is just not anything we've carefully characterized, but they do seem to escape a little worse. They're easier to catch. So, just at this level, we have this running around behavior. They seem a little, worse at escaping. So we're affecting a few extra things besides just the vocalizations, even though that's what we measured. But yeah, I don't know about the aggressiveness really. People have poised, there's like this review that suggests that the reason the PAG has been such a good locus for evolving new behaviors is that as poised in the midbrain, they have access to all these different motor modules. So therefore, it makes it likely that sort of a small change in the PAG can sort of couple things together and also redeploy things. Something that we didn't look at, given to an earlier question, sort of was asking like, what's the social response to this? I was also curious. We only had overhead camera, so we didn't measure it. Side camera would have been great, but sort of seeing mice also do this posture when they throw their head back and open their mouth and stuff. It would have been cool to look at how their posture also degrades in addition to this sort of temporal degradation, but it's not something we can measure too well from overhead cameras. But it seems like, I have the hypothesis that, singing mice have co-opted, singing mouse songs have co-opted USVs and then been redeployed. And part of this redeployment is, they make them when they're alone and they have this posture associated with them. So I think that is something that we could do in the future in regards to the evolution of this part of the brain. And then, yeah, like you said, with ability to have it associated with they also can't run around when they make songs. So there also is some sort of a rest signal that is associated with this sort of upright posture, I think, as well. That is another classical PAG sort of behavior, is freezing versus escaping and stuff. It seems like somewhere in the PAG, perhaps the immobility aspect of song has also been wired up together.
All right, thank you very much. I think it's about time, but yeah, thank you again for coming.
Great.
And
Yes, thank you, everyone.
Thank you.